We'll get started to stay on time. I want to welcome everybody to our dry bean production webinar series. And for those of you who joined us last year, we're trying to kind of continue along in our education about dry beans production and marketing and quality, and really keep building our knowledge around growing dry beans here in the Northeast. We have a number of projects and funders and partners that are engaged with us on this journey. And so Kristen Loria is here today with me from Cornell University. A member of Dr. Matt Ryan's team, and Sarah Pethybridge is our presenter today, also from Cornell University, and a member of our dry bean research team. I also just saw Ellen Mallory is on from the University of Maine, another team member working on dry bean research and outreach with our group as well. We have a couple of projects, project that Kristen and Matt and Ivy, who's also joining me here from UVM, have been working on, that's funded through the USDA SARE. And that's kind of where we started on our journey, working to develop and investigate dry bean production practices for organic production, and most recently funded through the USDA Organic Research Education Initiative to further that research with our partners that include Cornell, Maine, the University of Wisconsin, the University of Vermont, all of us working together with growers and end users to hopefully increase the acreage of organic dry beans here in the Northeast over the next few years. So with that, as I said today, Dr. Pethybridge from Cornell will be joining us and talking about disease management in dry beans, which there is a ton to learn about. But we also have a number of other webinars coming up. Here, if I can change my slide. We will be joined by Dr. Travis Parker next week from the University of California, and he's going to be talking about new dry bean varieties that he's been developing. And then on March 5th, we're going to have a dry bean farmer panel as well with farmers from our project that will be talking about their experiences and practices growing dry beans in the Midwest and the Northeast. So please continue to join us throughout the webinar series and keep learning about dry beans. So we do have some CCA credits available, and I will put up the QR code at the end of the presentation today that you can scan, or if you don't have those capabilities, please feel free to email us and we'll figure out some other options for you. And with that, I'm going to turn it over to Dr. Sarah Pethybridge, a plant pathologist from Cornell that works with beans, amongst other crops, trying to help us solve all of our disease issues that we deal with in agriculture. But today she'll just be focused on beans for the next hour or so. And if you could please, if you have questions, put them in the chat and we'll get to those at the end of the webinar. So with that, Sarah, I will turn it over to you.
Great. Thanks very much, Heather. I will just share my screen to get us started. Okay, can everyone see my screen?
Yes.
Great. Thank you. And thank you very much, Heather and everyone, for having me today to talk to you about one of my favorite topics, which is dry bean disease management. So today we're going to review just what do we mean by a disease and the impact it has on our dry bean crop. We're also going to talk about the different important diseases affecting dry beans in the Northeast. And we're going to split those up into bacterial diseases and fungal diseases and have a look at what crop growth stage each of those affect, and therefore what time in the crop production cycle we need to start thinking about each of these diseases and their impact on our crop productivity. We'll review for each of these diseases the symptoms so we can be more aware of them before the season starts. We'll also review the pathogen biology, so what drives the pathogen, how is it spread, and therefore the epidemiology of those diseases as well. So how do those diseases, how do those pathogens interact with the environment, and therefore when are they going to occur throughout the cropping season? But most importantly, we'll try and use that information that we've gathered to be able to improve our management strategies and prevent crop loss from these diseases in our dry bean crops. So to start with, what is a disease? What do we mean by that term? Well, it implies a deleterious effect on either plant growth or quality. Now, we can mean yield or crop loss, either through plant growth or quality. Now, in dry beans, it might be less beans being harvested, or the beans that have been harvested are of lower quality due to a disease or other means. So when we think about crop loss, we're thinking about reduced profits or direct losses in the crop that we're dealing with in that season. But we also need to think about the indirect losses. So when we allow disease to build up in our crops, we obviously also get inoculum buildup in that field, and that inoculum stays in that field and therefore poses a higher risk to disease when we grow a susceptible crop back in that field. So we're not only thinking about the direct losses in that season, but also the inoculum buildup and therefore the potential for crop loss in future susceptible crops. So you might have heard of this concept. These are the three factors required to obtain disease. We need all these three things to occur at the same time, and this is represented by the disease triangle. So we need to have a pathogen that is present, we need to have a susceptible host, the plant, and we need to have conducive environmental conditions. And they need to occur all at the same time, and therefore this triangle forms what we know as a disease pyramid. And these environmental conditions are going to differ depending upon each of the different diseases or different pathogens we're dealing with. For example, some diseases are exacerbated by warmer, humid conditions. Some are exacerbated by cooler, wet conditions. So the environment is really has a lot to do with when we might see outbreaks of disease throughout the cropping season and what type of season we're experiencing as well. As we know, that varies between years, especially in the Northeast. But overall, so we can be proactive and try to manage our diseases and try and prevent crop loss, we have to get an accurate diagnosis. So it's important to try and determine the organism that's responsible and know the symptoms of these main diseases so we can try and identify the appropriate control measures and have a longer-term plan for management strategies if we're planning on a rotation of dry beans in the future in that field. And of course, sustainability as well. We want to try and integrate methods that are going to be helpful within the cropping season, but also in the longer term to try and reduce the probability of disease and therefore the risk of crop loss. So in a way, we need to have some background information on our enemy, which is the different plant pathogens which affect our dry bean crops. And they may be grouped into their different types. So there's fungi which cause diseases, there's bacteria that cause diseases, there's also other diseases caused by viruses and nematodes, and also different types of pathogens. But the ones we're going to concentrate today and that are most important in dry bean production in the northeast are diseases caused by fungi and diseases caused by bacteria. So we're going to start off by looking at the different bacterial diseases that affect dry beans in our region. And in general, if you have a disease caused by a bacteria, they usually have initially very water-soaked type symptoms. The symptoms will, before you get to a brown or yellow lesion on the leaf or the foliage, you usually have these water-soaked type symptoms, either on the top side of the leaf or often on the underside of the leaf to start with. Now, bacteria can survive in the soil, they can survive on infested crop residues, but they can also be introduced by infested seed. And we'll talk about the different methods of dispersal for each of the key bacteria that are important in our region in a moment. But usually bacteria, once they're in the field, are dispersed by water. So rain splash, irrigation water, runoff water, and so forth. So this is in general what we might experience with diseases caused by bacteria, not only in dry beans, but in our other crops as well. So the first of the bacterial diseases affecting dry beans in our region is called brown spot. It's caused by the bacterium Pseudomonas syringae pathovar syringae. Now, the symptoms are quite distinct, as you can see here in this picture. The lesions on the foliage are usually quite small, up to about 1/8 inch in diameter. So they're quite small and they're usually circular, roughly. They're also, when they age, they tend to be brown or what we call necrotic in the center. And a common symptom of bacterial diseases is that they usually have this yellow or what we call chlorotic halo around the lesions. That can occur in some fungal diseases, but it's usually the norm with bacterial diseases. So on this bean plant, we've got symptoms of brown spot that are older. We have those brown necrotic centers and we have those yellow chlorotic halos. Now, often as those lesions age, and we can see this over here, the center of the lesion becomes dry and brittle, and it often falls out. So we tend to get this shot hole type appearance that you can see here in this older lesion. But brown spot can also affect the pods in addition to the foliage. So on the pods, you can see that there's circular lesions that again become brown or necrotic. There's not so much of the chlorotic halo on the pods that are more characteristic on the foliage. And although this picture is quite fuzzy, you could see that it may also deform the pods as well. So the disease in terms of yield will reduce green leaf area and therefore the ability of the plant to produce pods. It will also affect the pod quality and the bean quality as well within the pods. Now, from what we know about the biology and epidemiology of this pathogen and disease, we know that the pathogen survives in infested crop residues or seeds. It can also survive in the soil and in certain weeds as well, related weed species. It's got a generally pretty broad host range, but mostly other beans and dry beans are highly susceptible to brown spot. So it's usually these bean species, such as limas, snap, broad, soybeans, in the rotation that are contributing inoculum to our dry bean crops. So that information will be helpful to us when we try and design management strategies in a moment. So the brown spot is also favoured by warm temperatures. It's more of a tropical type pathogen, and it likes 80 to 85 degrees Fahrenheit, unlike myself. And it's spread among and between fields in wet weather by hailstorms, rain, and windstorms. So usually we would associate spread being quite small and localized with rain splash, and therefore generally within a field. But these windstorms and hail and rainstorms that we get, and perhaps more frequently in the Northeast in recent years, can also spread the pathogen between fields as well. Now, the second bacterial disease is called halo blight. Now, halo blight is caused by another bacterium of similar species. So again, it's a Pseudomonas syringae, but in this case, the pathovar, or the different type of this bacterium, is different. And in this case, instead of syringae, it's called Phaseolika. Interestingly, this disease was first reported in the 1920s in New York. I'm sorry about that. And again, it begins as water-soaked lesions on the underside of the leaves. And as the diseased symptoms develop with time, then they become more evident on the top side of the leaf. And we tend to get these very reddish brown lesions. And as we saw with brown spot, we also see these yellow rings or these chlorotic areas, which are usually distinctive for bacterial diseases as well, accompanying these reddish brown lesions. Whereas brown spot was more of a brown black, this one is more of a reddish brown. So the symptoms can occur on the foliage, and again we get these yellow to green halos around the lesion. They're quite distinctive and they're usually much bigger than brown spot. You can see that these are much more of a dramatic type lesion than brown spot. And interestingly, the symptoms can be suppressed at high temperatures. Although the pathogen is still there, we don't tend to get symptom expression and symptoms spread when it is at high temperatures above 85 degrees. Now, as infested seed is also a source of this bacteria causing halo blight, we can also get systemic infections of the seedlings from infested seed. And in that case, very early in the season, just after emergence, our plants might look deformed, the young leaflets especially become deformed, and they really become sort of a nondescript, chlorotic, generally yellow type appearance with no brown spots. So it's not, these symptoms look like a lot of other root rots that may also be causing damping off in young seedlings. They're certainly not distinctive for halo blight. So again, it's important to get these kind of symptoms identified by your local pathologist or local extension staff so we know exactly what we're dealing with, whether it's a bacteria or a fungi, which is going to dictate our different management strategies. Now, halo blight can also affect the pods, and on the pods, the symptoms are more of a circular, water-soaked type spot, but they can also occur as a streak along the pod sutures, along the outside of the pods. And often we also get bacterial ooze associated with these pod type symptoms. So that bacterial ooze for this pathogen might be sort of a whitey or a yellowy type appearance. I know it's delightful, but overall, this disease again is reducing green leaf area if it occurs on the foliage. If it occurs on the seedlings, it's reducing seedling vigor and therefore potentially affecting our plant populations. And on the pods, it's reducing probably the number of pods because these pods may upsize prior to harvest. And again, the quality, because these lesions do go to infect the beans within the pods. Now, just as we did for brown spot, we know a lot of information already about Pseudomonas syringae pathovar Phaseolika. Again, the pathogen survives in infested crop residues, seeds, soils, and weeds, just as the pathogen that caused brown spot did. Again, the dominant host in the rotation is probably beans because it's so susceptible. This one is favoured by cool temperatures. So instead of the hot temperatures that brown spot was exacerbated by, this one tends to be most severe when it's about 70 to 75 degrees throughout the season. And again, the spread is similar to the pathogen that was causing brown spot. Wet weather, hail, rain, windstorms within and between fields, probably adjacent fields. Now, the third disease caused by bacteria is called common blight. Now, common blight is caused by a different bacteria this time, not Pseudomonas syringae. It's caused by Xanthomonas axonopodus pathovar fascioli. So that word pathovar fascioli just indicates that it's specific to beans, being the genera Fasciolus. So again, this disease affects the foliage and the pods, but in general, the symptoms of common blight are a lot more dramatic and a lot more severe than those other two diseases that we've just talked about. So for common blight on the foliage, it generally starts as small water-soaked lesions, again on the underside of the leaves, but very quickly the lesions become brown or necrotic, and they become dry and brittle, and they tend to coalesce together rather than producing new lesions. So very quickly they can cause significant defoliation. Now, the symptoms usually occur in the interveinal areas, as you can see here, or the leaf margins, as you can see on this leaf as well. And it may cover very large areas of the leaf and again, completely defoliate the plants. Here you can see a focus of infection in our dry bean crop, which is causing significant damage. So again, this bacterial disease also affects the pods, and they're initially circular lesions, again, water-soaked lesions, and with age, they become a reddish brown and very much a sunken type lesion, whereas for the other bacterial diseases, they don't tend to have that sunken appearance. But they may also have, in humid conditions, yellow masses of bacteria associated with the lesions, sort of oozing out of the lesions, again, delightful. And if it's infected very early, it will also reduce seed quality as well. So in terms of how this disease affects productivity, again, it will be reducing green leaf area and therefore the ability to produce pods. Those pods are of lesser quality when they are produced and reduce seed quality, bean quality as well. Now for common blight, again, very similar epidemiology to the other bacterial diseases we've talked about. So the pathogen again survives in infested crop residue, seeds, soil, or weeds. Again, the dominant contributor of inoculum is other beans in the rotation. It also likes warmer weather, so it's more similar to brown spot than halo blight. And again, it's spread by rain and wind-driven rain as well. Now, this disease, bacterial wilt, we do not have in the northeast, but I just wanted to mention it because it's an extremely severe disease and we want to keep it out. So this disease is called bacterial wilt. It's caused by a different bacteria, Curtobacterium, I'm not going to pronounce that one, but it's generally in the central high plains or Nebraska type regions. And it causes wilting of the entire plant and plant death. Most often that wilting occurs in periods of water stress, so it's when it's high temperatures, over 90. And it's associated with very distinct symptoms of interveinal necrotic or brown lesions. It can discolor seeds if the plants do survive, but more often than not, the plants will die prior to getting to producing seeds. So it's usually reduces plant populations and therefore affects yield in that way. So in terms of bacterial disease management, I like to think of things in terms of strategies, things that we can do prior to planting to reduce the risk of disease in our crops to start with, and tactics or things we can do within the season to try and mitigate crop loss from the disease which is already occurring. So for bacterial disease management, we saw that all those pathogens were potentially introduced by infested seed. So it's really important to start with certified disease-free and or treated seed. So there might be more options for conventional dry bean production for treatment, more for fungal diseases, but using certified disease-free seed is the key to getting started, starting with a clean inoculum. Now, we also saw that all those bacterial diseases were most severe on bean species in the rotation because they were so susceptible, although the pathogens may survive on other hosts in the rotation. Beans were certainly the dominant source of inoculum. So crop rotation is going to be really important to try and reduce the inoculum to start with, and therefore reduce the risk of disease from the inoculum from within the soil, within the field. So we recommend rotating away from beans in that field for at least 2 years, specifically for bacterial disease management. And we'll get into more about that when we deal with fungal diseases more in a moment. It's also important to scout regularly. Some of these symptoms are not always obvious. For example, we saw that the water-soaked lesions usually first occurred on the underside of the leaves. So just walking through the crop might often not be sufficient. We might not pick up that very early symptoms of disease. But knowing what we're dealing with, trying to get onto it early to try and prevent the rapid spread of the bacterial diseases within the season is also going to be really important. Because if we're thinking of doing something within the season, so tactics, in-season control measures, our only real option is really copper-based sprays. Usually they're most effective for the bacterial diseases at mid-vegetative phase or early flowering periods. But I put a caution on that because it's sometimes when you apply copper in very high temperatures, which may coincide with early flowering, so above 85 to 90 degrees, that can cause some phytotoxicity to the flowers in dry beans, and we can get premature flower drop abscission. So be careful when applying coppers in early flowering. But we also need to ensure good spray coverage as well. The copper-based sprays are primarily protectants. So what we're doing is trying to protect the tissue that hasn't been infected already. So we're trying to prevent disease spread. And to be able to do that in sometimes a very closed canopy of dry beans is hard. So it's important to get good pressures and good nozzles to be able to try and optimize that variable. Now there's different OMRI listed coppers for organic production. So please check the labels of your different coppers. Some of the more common ones are Coside 3000O and Cuava as well. And in our trials, we haven't really seen any differences in the efficacy of the different coppers, whether they're OMRI or conventional or not, for bacterial disease control, but as long as they're used at the maximum labeled rate for the disease and for your crop. So check out the labels and use them at the maximum labeled rate. And when we're thinking about coppers, we have to also think about the restricted entry intervals. So it's usually 48 hours for most copper fungicides, regardless of whether they're conventional or synthetic products or they're OMRI listed type products. The restricted entry interval is usually that 48 hours. So again, you can find out that information by checking out your specific label as well. So moving on from bacterial diseases to diseases caused by plant pathogenic fungi. So the majority of plant diseases are actually caused by fungi, and they're generally microscopic and filamentous type organisms, and they often rely on spores to spread. Now, the fungi can be classed in two major lifestyles. The first is called obligate. So obligate fungi need their host plants their entire life, to a living host to produce their life cycle. And a good example of those are the powdery mildews. Now, other fungi can survive for certain periods without their host. So they're what we call facultative. So they can survive independently of the host by arresting bodies such as sclerotia, or resting bodies such as spores or different mycelia. This is an example of a facultative pathogen. This is white mold, actually in cabbage, but we'll be talking more about white mold in a moment, but in dry beans. So the different types of fungi dictate how the spores spread, and therefore what we know about the epidemiology of these diseases. And there's a great diversity. So for example, there's some fungi which spread their spores by wind. So they're producing these spores here on specialized hyphae or mycelia. But some fungi produce spores within these structures on the leaf surface. So here you can see these brown globose type structures with holes in the middle. And in those structures, there's spores being produced which ooze out onto the leaf surface in the right conditions. And then those spores are rain splashed, usually very short distance because of the fact that it's rain splashed, onto new plants to cause new infections. So there's a great diverse. Some diseases caused by fungi that are wind dispersed may spread further than others which ooze their spores onto the leaf surface and therefore rely on passive rain splash. So the first group of diseases caused by fungi we're going to talk about for dry beans are the fungal root rots. Now, there's a range of different root rots caused by a range of different fungi. And the first one is Fusarium. So the Fusarium fungal root rots are caused by this fungus, Fusarium solani, and this F spur simply says that it's specific to fascioli, so beans. So this root rot can cause a pre-emergent seedling death, so they germinate and they don't make it to the soil surface. They can also cause post-emergent seedling disease, so they emerge from the soil but just generally look weak, or they may die after that phase. And often it's that part that we actually see. So on those seedlings, when we dig them up, we might see red to reddish brown lesions on the stems and the primary root system, as you can see here. Those symptoms usually occur right at the soil line, but they can extend down into the roots further. And it usually occurs at about 2 to 3 weeks after planting. Within that, we get death of the primary and secondary root system. So we've got a greatly reduced root area, which is going to affect the thriftiness of the plants above ground. And the lateral roots above the infection may compensate to a certain extent. They may get going if there's sufficient soil moisture, but in general, your attainable yield from those plants has already been produced, and therefore you're going to get some significant crop loss already, even if the plants do recover. So the above ground type symptoms with this root rot, and all root rots, unfortunately, are fairly nondescript. So there's stunting, the plants just don't look right, and they look unthrifty. And also in general, chlorosis, or what we call the yellowing of the leaves. And that was very similar to our common blight type symptoms that we saw before as well with our bacterial disease. Now, here's some other pictures of fungal root rots caused by the fusarium. As you can see here, these reddish brown streaks that are occurring on the pointy end of these seedlings. You've still got some root mass below that being produced, but this will affect the water distribution of the plants from the roots up. Here, there's more severe symptoms. Now, again, we've got these reddish brown type symptoms, which are quite distinctive of fusarium, as opposed to some of the other diseases we're going to talk about in a moment. And although the seedling has survived, you've got significant impact on seedling development, and often you might get other fungi coming in and causing disease in that area as well. So from what we know about fusarium root rots, disease severity is usually exacerbated by abiotic stress. So if we have very moist soils, and if we have cool soils, so if we're planting into cool, moist soils that are usually less than 60 degrees Fahrenheit, we're of higher risk to fungal root rots caused by fusarium. Soil compaction will also exacerbate fusarium incidence and severity, probably as an effect on soil moisture and indirectly that way. But there's also other interactions that can exacerbate the damage because the plants just can't recover because they don't have a fully developed root system. So it could be other soil-borne pathogens, or it could be herbicides in conventional systems that are also weakening the plants, and they just can't get up and go. And again, the above ground symptoms are most obvious. If they do survive and get over that seedling stage, they may fall over at flowering when there tends to be higher temperatures, or early pod set when there's more stress on the plants because of that phenological change and usually that higher temperatures. Now, we also get fungal root rots caused by Rhizoctonia, and Rhizoctonia solani is the fungus we're dealing with here. Again, it usually causes nondescript damping off, so it's hard to tell from the symptoms which fungal root rot is caused by what fungus. And we tend to get very linear, circular lesions on the stems with Rhizoctonia. And rather than that pink brownish type lesion for fusarium, we tend to get very reddish to red brown and sunken lesions, as you can see here on this seedling. Now, these lesions usually tend to encircle the stem, and in most cases will girdle the stem, and therefore cause considerable stunting of the developing seedling. But we tend to also get root rot and stem rot, and because of that, girdling stem cankers, but it can also cause other diseases on the dry bean plant as well, such as pod rot, and we'll see more about that in a moment. So nondescript stunting type symptoms, usually associated with pre-emergent seedling death, damping off, rather than post-emergent seedling death, and usually this reddish to reddish brown sunken type lesions, which often girdle the developing seedling. So for starters, we tend to get that seedling death, and here you can see some classic symptoms of lesions caused by Rhizoctonia on these developing seedlings. So they tend to occur slightly higher than right on the soil line. But we can also get root and stem rot in more advanced plants. We can get stem cankers and pod rot. So it's quite a diverse pathogen, really. To start with, it can cause a foliar aerial blight, as you can see here. So again, pretty nondescript type symptoms. It could be a range of different abiotic stresses as well. So it's important to try and get it diagnosed and to figure out exactly what you're dealing with. On the pods, it usually occurs when there's direct contact with moist soil. And so these are some of the symptoms associated with pod blight. So here you can see somewhat water-soaked lesions but generally brown flecks occurring over the pods. Not really, when we compare it to the bacterial diseases, there's no real yellow chlorotic halo that we would associate with bacterial diseases. So just this brown necrotic lesions associated with pod rot caused by Rhizoctonia solani. But we can also get seed rot or bean rot as well. And often this occurs after we've stored the seeds for a while as well. So here you can see this white mycelia that's being produced on these infested seeds. And if we look really closely, you can sometimes see these black to brown sclerotia, which are the resting body of the fungus, Rhizoctonia solani. So Rhizoctonia is a facultative fungus. So that means it can use these resting structures to survive with periods without a host. So again, if we try and preserve our thoughts about management to strategies that we can use before planting and tactics that we can use within the season for root rot management. Root rots, the fungi that cause root rots, are not seed-borne, but they are soil-borne. So crop rotation is one of the most important tools or strategies that we can use to try and mitigate the risk of crop loss from root rots. And again, we recommend rotation away from any type of beans, so soya beans, snap beans, dry beans, for at least 2 years in fields. Now, to a certain extent, we can get some control from fungicide seed treatments for root rot management. And we can also apply, if we're conventional, different in-furrow treatments as well. And the most effective fungicide for Rhizoctonia control is azoxystrobin. That's the active. The product name is Quadris. But again, if you're conventional, please check local registrations to make sure it's registered for dry beans in your area. So fungicide treatments will give you some protection, but these in-furrow treatments, if you've had a problem in that field before with Rhizoctonia, in-furrow treatment of Quadris will be quite effective. Now, there are a range of different microbial biopesticides. All of these are OMRI listed for the control of root rots as well, and they're soil applied, so generally within the furrow. Now, we don't have a lot of information on the efficacy of these microbials for the control of root rots, but they are listed as having sub-control. So there's some different products here, Actinovate. So that contains a bacterium called Streptomyces lidicus, and that acts on different fungal root rots by producing toxic metabolites and producing competition for the Rhizoctonia or the Fusarium in that area. There's also another one called Root Shield, which is a trichoderma species, so another fungus, and it enhances stress tolerance of seedlings, and it also suggests that it promotes the growth of the seedlings, so it will try and outgrow the plant pathogenic fungi. But there's others such as Soil Guard, which is another different type of fungus, another Streptomyces called Mycostop, different trichoderma called Biotam, and Pre-Stop, which is another type of fungus. It's now called Clonostichus. So there are microbials out there. We do need more information about their efficacy to be able to recommend specific ones for root rots for dry beans. So for root rot management, just as for bacterial diseases, it's also really important to scout and be out in your fields regularly, as I'm sure you are, and try and be proactive. For example, if you're seeing areas of Rhizoctonia folia blight, that may lead to pod blight, or if there's areas of lodging in the field, that may also lead to areas of pod blight because of contact with moist soil. So for root rot management, it's important to be aware of the and be aware of the optimal soil moisture conditions for planting. So trying to avoid planting temperatures and moisture conditions that might exacerbate losses from root rots is going to be the key. And in general, cold, wet soils generally exacerbate losses from root rots, either by pre-emergent seedling death or post-emergent seedling death. So also the soil conditions in the field will also have a significant impact on the incidence of root rots. So trying to avoid compaction in general from a farm systems level, and also trying to promote good practices to provide a vibrant, microbial active soil to improve soil health is also going to be important for an integrated disease management approach. Now, the second group of fungal diseases that I wanted to talk about today is called anthracnose. Now, anthracnose can again affect the foliage and the pods. It's a fungal disease caused by the fungus Coleototricum lindamathium. Now, the symptoms of anthracnose are quite distinctive, and it also can be an extremely destructive disease in dry beans. So to start with, we tend to get these rust brown to black lesions on the foliage. They tend to follow leaf veins, as you can see here, or veins on the petioles, and they first do that on the underside of the leaves. So you turn the leaf over and you seem to get these brown patterns following the veins on the leaves. But those tend to rapidly develop into round, sunken eye spot lesions on the top of the leaves. But those lesions also sometimes might have a pinkish type appearance, and that is because the fungus is producing masses of spores on those lesions to help spread and disperse that fungus and cause more disease. So those masses tend to be more of a pinky orangey type of mass. And together, this disease can cause extreme defoliation, can also kill plants outright as well. So on the foliage, it's reducing green leaf area and therefore crop loss in that way. But it also directly infects the pods. To start with, anthracnose can induce premature flowering, and therefore we can get pod abscission earlier, and therefore we've reduced that marketable unit, harvestable unit, which are the pods. You tend to get these quite spectacular lesions on the pods, very brown, black, and they tend to be sunken. Again, there's no yellow ring around them, which was the case for bacterial diseases. And they tend to have this orangey mass, as you can see here, orangey pinky, and that's the mass of spores associated with the fungus. So in this case, those lesions have gone directly through to the internal part of the pod and started to infect the beans. This is what it looks like when those hulls are dried out. Again, it's quite distinctive. It tends to have a targety type appearance to it when it dries out. But they can also infect the seeds directly as well. And on the seeds, it causes discoloration and deformation of those seeds. And really, infected seed is a major means of introduction and dispersal. So there's one key so we know about pathogen biology that we can use to our advantage for disease management. That is using clean, disease-free seed to initiate our crops. Now, the Coleototricum lindamathium has different races as well that cause anthracnose. So we can get different varietal susceptibility as well. Now, we don't know a lot about the different races that occur in the Northeast, but we certainly do within the Michigan and across the plains areas. And this is the same race which has been inoculated into two different cultivars of dry bean. One is obviously quite resistant and one is extremely susceptible. So once we gather more information about the races of Coleototricum lindomathium in our area, that will give us more information about which cultivar to recommend as being more resistant to anthracnose in the northeast. Now, this is the life cycle of the fungus that causes anthracnose. So it survives and overwinters in infested crop residues or debris and seed. So seed is again the most important primary source of inoculum. And once we get disease, the disease is the spores are spread by rain splash, and that causes new infections and disease spread, and the cycle keeps going. So we get multiple infection cycles per season. So again, the spread is by rain splash or windstorms, so usually within the field or between fields if we get those storms. It likes high humidity, so above 92%, but it's got a fairly broad temperature range. It's happy whether it's 55 or it's happy up to 80 as well. But overall, it's spread and exacerbated by frequent, moderate rainfall. So again, if we try and think about anthracnose management in this way, using certified seed is the key for anthracnose management, to try and keep it out to start with. If it's fungicide, it could also be fungicide treated. A lot of those fungicides will be effective for the Coleototricum. If it's conventional, our options are more limited if we have an organic production system. But the key for anthracnose management here is a crop rotation to cereal or corn. Because the Coleototricum lindomathium can survive on a range of different weeds and a range of other crops in our rotation, the key will be to rotate to cereal or corn for a minimum of 2 years, which was also the case for our bacterial diseases. Now again, Coleototricum will survive in crop residues, so it's important to try and promptly encourage decomposition after the crop is harvested. So often we incorporate or just cover it with soil to try and encourage that decomposition. I know that's hard in a no-till system, so that will depend upon your particular production system. But overall, host resistance could also play a part in the management of anthracnose. It will probably be variable in the degree of disease suppression and the durability of resistance. But what we often on seed packages, you'll see these two different terms, tolerance or resistance. So tolerance means that, yes, the plant or cultivar may become diseased, but it's unlikely to have a significant effect on yield. Whereas resistance can mean a range of things, and it can mean a range of different levels, but usually it doesn't mean immunity. So the extreme of resistance would be immunity. So when reading seed catalogues, it's important to try and differentiate those two terms, tolerance and resistance. We do know that Eclipse, one black bean variety, is resistant to anthracnose, but because of the lack of knowledge about races to Coleototricum lindomathium in our region, it's important to try and gather local information to be able to select cultivars that are going to be more resistant in your region because of those races and the change in races over time. Now, the last disease I wanted to talk about today was white mold, one of my favourite diseases. So white mold is caused by the fungus Sclerotinia sclerotiorum. One of the hallmarks of Sclerotinia sclerotiorum is it has an extremely broad host range, so not just beans, in this case, like our bacterial pathogens. And because of this, it's considered endemic within our northeast specialty and field crop rotations because it survives on weeds and a range of other crops as well. So getting back to what we talked about with direct crop loss and indirect crop loss, direct crop loss for white mold means less pods because they've become diseased, and often they upsize, so they don't even get to the harvest. We could also have a reduced number of beans per pod as well. So if they do get to be harvested, there'll be less beans. And we could also have product contamination if they do get harvested. So here you can see that there's some cranberry beans, and these black structures that have been harvested along with them are the sclerotia, which are the resting bodies of the fungus. So this one again is a facultative plant pathogenic fungus. So it can survive in the soil in the absence of a host as these sclerotia within the soil. Now, when we think about indirect crop losses from white mold, we think about those sclerotia again. So those sclerotia, which are on the diseased plant parts, fall back to the soil surface, and they form the soil-borne inoculum for the next successive susceptible crop. So in that way, we've increased the risk of crop loss from white mold in future susceptible crops. So again, we have the direct loss and the indirect loss from the soil-borne inoculum. Now, white mold symptoms, you may already be familiar with these because they're quite distinctive, is that usually they start off as a bleached light brown or necrotic lesion, as you can see here on the pod, and very close after this development of these lesions, very rapidly we get the production of cottony white mycelia. Now, a lot of things have mycelia, but this one is quite a distinctive mycelia. It's cottony and white, and you can see that it tends to ball up into these cotton balls. And that is the start of the production of the black hard sclerotia, which is the soil-borne inoculum. And you can see that there's some sclerotia just being produced here. But bleached lesions, cottony white mycelia, which is the start of those sclerotia. So they may occur on the pods, but they may also occur on the stems as well and cause crop lodging that way. So again, they're bleached necrotic lesions that you can see, and we get signs of the pathogen, that cottony mycelia and sclerotia. And often if that's the case, when you're walking through the crop, things just might not look right at the top of the canopy, what I call flag leaves. So if they're wilted type looking leaves, often you part the canopy and look down towards the soil, and you might see symptoms of white mold on the stems and signs of the pathogen that you can see here. So it tends to have this warning sign at the top of the canopy. But there's other diseases of pods out there as well, which can cause crop loss. This one is one of the more common ones. It's called grey mold, and it's caused by another fungus, Botrytis cinerea. It's kind of got that grey, fuzzy type look, which I see on my strawberries in the fridge, because it's the same fungus as well. So quite different from white mold. These necrotic pods, they tend to be shriveled, and this grey, affectionately known as fuzzy mycelia. Now for white mold, if we can get back to the disease triangle, because the pathogen is usually sufficient in sufficient quantities to cause disease because it's of endemic nature within our rotations, we usually have enough pathogen in our field. Now, we usually have also susceptible plants because there's no significant resistance or immunity to white mold. So it's usually regulated whether we get a white mold year or not by the environment. And it's usually the dominant influence of the conditions in spring and summer, and we'll take a closer look at that. So here, these are the conducive factors which cause white mold. So we have to have moist soil. We need to have temperatures less than 30 degrees because I'm still in the British system. So that is about 80 degrees. And we have to have a dense canopy, and all those things tend to be correlated. So we tend to, these conditions are conducive for the germination of sclerotia into what we call apothecia. Now, this is the life cycle of Sclerotinia sclerotiorum. So we start off with this primary inoculum, the sclerotia in the soil. Then they germinate into those apothecia. So these apothecia are those small mushroom-type structures. And on the top of these apothecia, we get the production of ascospores, which are these small colourless type balls. And they're liberated into the wind and spread by wind. Now, an interesting feature of the life cycle of this fungus is that it infects only the flowers. And they can use the dead, dying flowers, once it's colonized the flowers, as a nutrient source to be able to infect green tissue, like the pods and the stems. So it has to infect the flowers first. It can't infect the green tissue directly. So immediately by knowing that, we know this is an intervention point we might be able to use to our advantage for disease management. So once it's infected the flowers, we then get our typical white mold symptoms, bleached lesion, and our signs of our pathogen, the mycelia and the sclerotia. Now, the sclerotia which contribute to white mold epidemics in the field are usually within the top 2 inches because they have to be able to germinate and push their way to the surface to be able to produce these small mushroom type structures, as you can see here. They're usually 1/8 to 1/4 inch in diameter. They're extremely small, and because they're not pink, which would be nice, this small off-coloured brown, it's very hard to find them when crawling under a dry bean canopy to try and scout. But in general, they like soil moist, shaded and cool conditions, 40 to 60 degrees under the canopy. But usually it's only the sclerotia within the top 2 inches. And that's going to be important more in a moment. So for our own research, we produce spores in the lab. We produce our sclerotia on rotten vegetables of various kinds. And then we put them in these cheesecloth bags and we place them in cool water in the fridge. And we aerate the water with like a fish tank pump. And when it's been sufficient time for them to what we call condition, which means that they're able to germinate and produce the apothecia, we place them onto these Petri plates on sand, and then we keep them moist. And then hopefully under our right light conditions, they will germinate, produce their ascospores, and then we can suck off the ascospores with a vacuum pump and impact the ascospores onto these filter discs. And the brown powder-like substance that you see here are the ascospores that we've collected. Now, these filter paper discs can be stored in the freezer, and when we can make suspensions for inoculum to be able to do trials for our research for white mold, such as evaluating different products, different OMRI listed products, for their efficacy for white mold control. But we know from the biology of the pathogen that the inoculum for white mold epidemics generally occurs from the field that we're dealing with. So it's predominantly from within the field. It doesn't usually blow in from the field next door, and it doesn't usually blow in from the valley down the road. This is because when the spores do escape the canopy, they were colourless, as we saw in the picture. So they're extremely susceptible to degradation by ultraviolet light. So the spores, if they do escape the canopy, they're not usually fit to infect by the time they get to another location. So what we do know is that one sclerotia leads to many ascospores, which means leads to crop loss. So in dry beans, we only need one sclerotia per linear metre of beans to get significant crop loss. So it's a very low level of sclerotial inoculum that has to be feasible if we're going to manage the sclerotia only to try and control white mold epidemics. So again, we're thinking of this in terms of strategies and tactics, this time for white mold. So if in terms of strategies, we could plan our crops with different agronomic variables, such as wider row spacings, we could decrease plant populations, but all those things are counter to having higher plant populations, more significant canopies, green leaf area to be able to produce higher levels, higher numbers of pods. So it's counterintuitive to producing high yielding crops. So everything has pros and cons and different trade-offs. We could also try and manage weeds most effectively. So often if we manage weeds not only for crop rotations, because Sclerotinia, Sclerodiorum will infect many of our broadleaf weeds, but also to try and produce airflow through the canopy. So weeds, especially within our inter-row areas, will try and block that airflow, increase our relative humidity, and extend our leaf wetness, which may be more conducive to infection by our ascospores and disease development. And again, the same principle here is used for upright varieties to get more airflow through that canopy. Now, also in terms of strategies, the main rotation strategy is rotation to the non-hosts, which is corn and grain. And we're going to talk more about sclerotial survival in a moment. But for white mold, we recommend at least 3 years between susceptible crops in fields. And again, also if we're going to rotate to our non-hosts, such as corn and grain, also controlling those susceptible broadleaf hosts so we don't get perpetuation of the inoculum on those hosts while we're rotating and trying to get the benefit of a rotation to a non-host for white mold control. Now, we know that sclerotia will survive in seed and it can be harvested with infested seed. So using clean certified seed, again, is the key to not only anthracnose management, but also dry bean management, white mold management as well. Now, the next question is, how do we deal with the residues and in terms of sclerotial management as well? So if we've had a crop that's had a significant epidemic of white mold, what's our best bet to try and manage the sclerotia and reduce their probability of living until we get to a net susceptible host? Well, we've done some trials in Geneva to try and answer that question. And this was established in fall 2020. We placed 30 sclerotia in mesh bags and they were sewn closed and they were deployed as pairs. So the mesh bags were either put on the surface or they were slightly buried. So it's similar to a low tillage type system, so buried at around 2 centimetres. And we sampled them on 24 different sampling dates over nearly 2 years. And we had 4 replicates of each treatment. And the objective here again was to quantify the survival time for those sclerotia in New York conditions. There was some information already from the Midwest, but we wanted to know more information about our local inoculum survival. This is what our experiment looked like when we deployed the different cages of, you can see the mesh bags with the sclerotia within them across our field. And these are our results. You can see here on our y-axis, we have the percentage of sclerotia recovered compared to our date on the x-axis. And you can see that we either recovered from the surface, 0 centimetres, or just below at 2 centimetres. And we can see that we have more intact sclerotia retrieved. So this is just the sclerotia retrieved, not that they were viable or not, from the surface than at 2 centimetres. So there's more likely to be a faster rate of degradation of sclerotia when they were placed just under the soil compared to on top of the soil. Now, this is sclerotial viability. So the viability of the fragments that we recovered. So again, sclerotial viability percentage on the y-axis versus time. And we can see that we have more rapid decline in sclerotial viability at that 2 centimetres than at the surface, and that there was less viable sclerotia at 2 centimetres than at the surface. So overall, our recommendation is do not leave sclerotia on the surface if you can avoid it. If we can cover them with soil, we can try and promote their degradation, presumably because we have increased surface area for microbial degradation. So in a healthy soil, it will hopefully do its thing at a faster rate if we cover the sclerotia with soil. Now, in our trial, we had less than 20% of sclerotia remaining after 18 months. So again, we recommend a 3-year rotation between susceptible crops, mostly because of that very low threshold in sclerotial inoculum density. You only need 1 sclerotia per linear metre of dry beans to get significant crop loss. We've also done some experiments looking at the role of cereal rye, and specifically rolled crimped cereal rye, for white mold control in dry beans and soya beans. So here we again know that from the biology of the pathogen that the sclerotia within the top inch of that soil profile or so forms the inoculum for white mold epidemics. And because of the interest in this rolled crimped cereal rye system, and particularly for organic production of soybeans and dry beans, we wanted to investigate the effect of that rolled crimped cereal rye system. What we found was that rolled crimped cereal rye barrier on the soil reduced the incidence of white mold overall. And that was because we had a reduced incidence of what we call apothecial germination. So germination of these apothecia, which produced the spores. But we also found an increase in the non-functional germination. So non-functional germination was defined as when the sclerotia germinated, but didn't form that round disc. And if they don't form that round disc, they therefore don't produce ascospores, don't produce inoculum to the crop, and they also don't germinate again because they've just exhausted all their energy within that sclerotia. So we believe that this effect would also have the effect of reducing sclerotial inoculum over time, as long as we don't bring new sclerotial populations from deeper in the soil profile with tillage. So it's a way of purging that upper soil profile of sclerotial inoculum. Now, in terms of management of white mold with resistance, we know that in dry beans and other crops, resistance is controlled by multiple genes, and there's really no strong resistance in our varieties. But what we can say is we recommend using more upright varieties to get more airflow through the canopy to reduce the probability of infection and disease development. Now, if we're thinking about in-season control of white molds, if it looks like it's going to be a white mold year, cool, wet conditions, and we've had problems with white mold in the past in that field, in susceptible crops, we do have a range of different options for conventional and organic production for in-season sprays. So these are the selected, what we've selected, the most efficacious fungicides registered for white mold in dry beans in New York. So I again encourage you to check on your local registrations and things like local rates, local PHIs, according to your labels in your region. But the gold standard on this list we consider for white mold control is Endura, followed by Amiga, or they're fairly similar in their control, whereas Topsin is also very effective as well. But we've had less success with Proline and Switch in New York, and for Rovril and Preaxor and Approach. But in other regions, such as the Midwest, they've had very good control of white mold, but more in soybeans as well. So perhaps there's some differences in sensitivity within our pathogen populations to different fungicides, but in our trials, we've had excellent results from Endura, Amiga, and Topsin. But I think probably the most important critical factor for white mold control using in-season sprays is getting that timing right. So it's critical that the timing of the first application be with early flowering. So typically for in-season sprays for white mold control, you might put on 2 applications. Usually the first is set at 10% flowering. So that is 10% of plants with at least 1 open flower. So that's really early in flowering because we need to protect, most of our fungicides are protectants primarily, those who protect those infected blossoms to serve as an inoculum source. This is our only period of intervention for white mold control, protecting the flowers from ascosporic infection. It's also important to know that with our in-season sprays, they're mostly protectants again. They therefore have limited systemic movement. And if they are going to move, it's usually only upwards in the plant. So therefore, especially in a dense canopy such as dry beans, adequate coverage is important. So thinking about flat fan nozzles and high or sufficient spray volumes is going to be important at getting the target, where getting the spray and protect those targets, which is the flowers in our dry bean crops. Again, there's a range of different OMRI listed products for white mold control. A lot of them, again, are protectant, so they're not curative. And they usually act on contact activity. They're usually best applied preventatively before rain, and the reapplication schedule is usually a lot tighter than for conventional fungicide. So usually you might put it on at a weekly schedule rather than perhaps a 10 to 14 day schedule for a conventional fungicide. And it's also important to note that OMRI listed products are registered, but efficacy is not a requirement of registration. So perhaps check with researchers and extension staff in your area for local efficacy data. So biopesticides are defined by the EPA in 3 different groups: microbials, biochemicals, but there's also this 3rd group of plant-incorporated protectants, or GMOs. And we're just going to briefly cover the first 2 of these, microbials and biochemicals. Again, most of these, but not all, have suitable for organic production. They're all contact, and some induce systemic acquired resistance as well. So the argument for use of biopesticides in not only organic, where they have to be used, but also for use in conventional, is the efficacy. A lot of them do have good efficacy data. In some cases, other disease management strategies may be incomplete, so we may have run into fungicide resistance and we're trying to prevent it or manage it. We're trying to maybe protect beneficial organisms, so trying to reduce the off-target effects. Oftentimes, they have shorter restricted entry intervals than some of our other products, like our coppers, of 48 hours. And they might have shorter pre-harvest intervals as well. And of course, there's generally higher degree of worker safety compared to our synthetics. Now, microbials may work in a range of different ways. Firstly, they may be antagonistic to our target. So they just may produce a toxic metabolite, for example, such as down below, or they may just outcompete and parasitize the target that we're dealing with. It may also be competition, so it's just outgrown the target in that area. Toxic metabolites, so it's directly toxic to our pathogen, or it might promote growth or stress tolerance in our host plant, or we might induce resistance. But the common ingredient in a microbial biopesticide is a microorganism.
Hey, Sarah, I just wanted to give you a 10-minute
Yep.
warning. It's
Sure.
120.
I will race through the next.
Sorry.
Thanks, Heather.
Yeah.
So here's some examples of those microbial biopesticides that are commercially available, and it's obviously non-exhaustive. And one of the two that we've found to be quite effective for white mold control is double nickel and Stargus, which is a Bacillus amylocofaciens, but different strains. And some of the features of those is that they have spores, so they're very durable for storage. Now, Lifeguard is a common biochemical biopesticide, and that induces systemic acquired resistance. And the other microbial biopesticide I wanted to mention for white mold control is one called Contans. It's a fungus called Conotherium minitans, and it's a fungal parasite of Sclerotinia sclerotia. But to be effective, it needs to colonize the sclerotia directly, so it needs to make direct contact. And therefore, it's probably better to spray onto infested crop residues before incorporation, because the target is right there and easier to get to. So the theory is that these degraded sclerotia will not produce apothecia and therefore produce ascospores. Now, there's also a range of different biochemical biopesticides listed here. We don't have a lot of efficacy data for white mold control with these products. And the same is for these products as well. But again, I just wanted to mention them because they're common occurrences in terms of biochemical biopesticides. The other acceptable products for organic production are these different copper compounds, and you may also find a sulfur or a mineral oil, which is acceptable for OMRI production. But it's also organic production, but it's also important to ensure the surfactant or the spreader you're using with any in-season spray is also OMRI listed. And again, the keys to success with any of these products for organic production is to have local efficacy data, be proactive, ensure coverage, check things like tank mixing and check the water pH, and check the expiry information on the product. Now, just some brief information about product efficacy from our trials with white mold. Again, we've had our non-treated, the incidence of pods with white mold is quite high. All of these products have been quite effective at reducing disease incidence, but especially double nickel at either of our rates, and similar to, not significantly different to, commercial fungicides here. Again, in this trial, we had 2 different rates of double nickel, 3 different rates of Stargus, and our conventional pesticides. And again, they were very similar in their efficacy, not significantly different, and they did a great job compared to our non-treated. But again, getting back to the timing, we've found that consistently in our trials, whether it's conventional or our microbial biopesticides, applying them early and rather than missing that early flowering window is really important for their control. So overall, we recommend the Bacillus amylocofaciens strains for white mold in-season control for both conventional and organic production, because their efficacy can be analogous to conventional production. Some of the commercial products are double nickel or Stargus. And overall, there is a complex of diseases affecting dry bean productivity, from bacterial diseases, root rots, anthracnose, white mold. But don't give up. All is not lost. Hopefully we can do an integrated disease management strategy for these diseases, such as certified disease-free seed, scouting to get a proactive approach, using a systems approach like planning the rotations, using host resistance when it's available, being prepared for in-season sprays, and trying to have a healthy microbial active soil. So in conclusion, thank you very much for having me. I'm happy to answer questions.
Great. Thanks, Sarah. That was great. That is So much good information on all the diseases that
Yeah.
bean growers have to deal with. We should have broke this into like six webinars just to like break it to people gently.
Yes, that's right. Yeah. Sorry, everyone.
Yeah, there are a couple of questions. One question was asking about spacing and planting that may help reduce the occurrence or spread of diseases. Is there, and I think you touched on a little bit, but
Yeah, I mean, we haven't specifically done spacing trials or plant population trials, but I guess it's a balance with yield potential as well, and probably the planting equipment that you've got as well that's going to be most practical. I mean, we
yeah.
tend to plant on a 30-inch rows or a 24-inch rows. What do you guys do, Heather?
Yeah, we use 30-inch rows. I think at least most of the growers organically that I know use wider rows because they're also cultivating weeds, so
Okay.
kind of have a bigger spacing, but yeah.
Yeah.
Somebody wanted to know if pollinators play a role in the spread of white mold and maybe other diseases too from flower to flower. Has anybody looked at that?
Yeah. I mean, there's been some studies using thrips, for example, that have been able to transmit the spores or drier spores of certain fungi, thinking of Staphylium leaf blight in onions. But there's not been any studies with regards to anthracnose or white mold. With those other spores that have been shown to be effectively insect transmitted, there have been darker spores, so they survive ultraviolet light longer, whereas a lot of them, like anthracnose and white mold, are colorless, and they tend to break down within half an hour to an hour with exposure to sunlight outside the canopy. They're still effective at causing disease.
Yeah. Very
Yeah.
neat. There's just one last question about seed treatment options. Somebody said you have some great seed treatment options also that are not mentioned that protect seed at planting.
Yeah, so this is conventional.
It doesn't say, but
yeah, so we tend to use some control for the oomycete type fungi, such as a metalaxyl type ingredient, but also a fludioxonil type component. It was called Maxim. I think it's still called Maxim. Thiram may also be effective for the Coleotrichums, but I'm not aware of any effective OMRI listed products. And actually, we're working on that for infested seed for our Coleotrichums for anthracnose within the grant that we're working on.
yeah, so more to come.
Yeah.
In the future. Well, that's all the questions today, and people are just saying thank you very much.
Thank you.
Just so folks know, a few people have asked if this will be recorded or if the recording will be available, and yes, we'll make it available on our website. And there's also a new website for our USDA OREI project, and we'll make sure that it gets on the new website as well as our UVM website. So with that, thank you again, Sarah, for your time today and all the great information.
Thank you for having me, everyone.
Yeah, just looking in the chat, but otherwise, oops, I am sorry. For those that needed CCA credits, I need to put that up for you, and I will do that for those that are waiting. Sorry about that. If you need to scan and get your credits, there they are, 1.5 credits for today in pest management. And we will see everyone hopefully again next week when we hear from Travis Driver from California and his development of new bean varieties for the dry bean market. So thanks, everyone. Have a great day.
Bye, everyone.
Bye.